So today is Monday, the 11th of May, our last formal get-together class for Bio 2510. And what I will do, I guess, as I typically do, is open up the floor for questions related to maybe lab stuff first, and then we can talk about lecture stuff. Do have the lab practical scheduled tomorrow, Tuesday the 12th from 2 to 6. It'll be open in Blackboard. I sent you guys a reminder on that. And I think I also said that you'll have access to your results. Did I say like Thursday? I'm trying to remember the time, day. It's in that email.
Thursday at noon.
Yeah, thanks. That's what I thought. Okay. And then also we've got our last exam scheduled for Thursday the 14th. And that was going to be open from 6 p.m. to 10 p.m. Right? Over chapters 15 and 16. And there'll be a slide at the end of today's PowerPoint that will review all this too. That will remind you of all that. But I wanted to mention it while I thought of it. Any questions on the lab stuff? All right. How about chapter 16 questions? We got into the lymphatic system last week, I think. And anything you'd like to ask about or review?
And I'll just post it in the chat, a question.
Oh, thank you. Oh, those chemicals, I would say pretty much sort of dissipate in the body. You know, they're pretty much used to, well, depends upon what type of interferon it is. There's like, gosh, at least 7 or more different kinds. And I think we mentioned last week, let me see if I can find that slide. You guys see the functions of lymph nodes up there?
Mm-hmm.
Okay, great. We talked a little bit about, let's go to this one slide that summarizes all of the innate defense mechanisms. There was that video that, I'm not sure if we watched it or if I told you to watch it, that specifically talks about the role of interferon in innate defense as it related to cells that were being invaded by viruses. But as we'll talk about today, when we get into specific defense, they're also involved in kind of firing up what are called B cells. So back to your question, what happens to them? You know, once they've done their job, either in control of viral infection, by warning other potential cells that, hey, virus is going to try to attack you, or whether they're helping to stimulate B lymphocyte action, they're not really being produced or secreted after the battle sort of has been won, so to speak. So their concentration just sort of, I would say, kind of tails off in the body. It's there as a chemical to get immune cells fired up, basically. And then once they've been fired up and stimulated and so forth, then the level of interferon really begins to decrease. But I suppose if you monitored your blood interferon levels, you could see it spike and so forth, depending upon what point of the infection you're in, so to speak. So that's a good question.
Want it? Food?
Okay, and you're writing here, okay, and typhoid is listed as a virus and bacteria. Oh, typhoid is listed as a virus and bacteria. Where did you see that? Listed under both on the PowerPoint. I don't know. You got to tell me where you saw that. I guess I'm just not visualizing that for some reason. I could see it would be listed under bacterial, but I'm not sure about virus.
On the pathogen like chart.
Oh, okay.
Bacteria, fungi, protozoa.
Yeah. So let's go to that. This one. Typhoid. Oh yeah. I don't know. I'd have to do some research on that, to tell you the truth. I would have listed it under bacteria, but I'm not sure why it's under viruses. I don't know. Stay after class and I will grab my microbiology book and I will tell you. Other questions on stuff we talked about last week? I think we got about as far as this slide. And I know we kind of ran out of time, but there was this list of innate defense systems. Oops. And this is certainly something worth looking at, this table 16.3, and the accompanying videos and hyperlinks. You're going to want to spend some time looking at this table. So again, the takeaway message is all of these defense systems that you see listed are there regardless of how many times you may have gotten the virus, a virus or a bacterium or any sort of pathogen. Your body has these built-in systems to negate any and all possible pathogens, with a pathogen being again defined as an entity, bacterium, virus, protozoan, fungus, that could cause infection and or disease. Okay. So again, spend some time on this table. What we want to talk about today is the second part of sort of immunity, and that addresses what do we have going for us, if you will, when we target a specific pathogen. How do we build up memory to that pathogen so that if we're exposed to it later in life, we can nip it in the bud without it causing us to get sick like it did the first time? That's basically what specific or adaptive defense is going to be talking about. And so most of the time when we talk about this, we use the term immunity. I think my microbiology book, in fact, also puts immunity in this first category, if you will, under innate defense. I personally put it in the second because when I think of immunity, I think of specific defense or adaptive defense. But you could make the argument that these non-specific barriers that we talked about, first, second, third lines of defense, is also part of immunity. It's part of protecting ourselves. Our immune system protects us from foreign invaders. And as I said a minute ago, we're going to be focusing on this unique ability of our immune system to recognize that pathogen the first time and to build up systems such that if we get it later in life, if we get exposed to that same virus or same bacterium, we can knock it out before it can cause us an infection. And just FYI, I don't know that I can access chat as I'm going through the PowerPoint here. So if somebody is chatting or has a question, just interrupt me. And if you guys have any questions as I go through this, please don't be afraid to speak out real quick and say, hey, what's this about? Or could you review this or whatever? Everybody okay with that?
Yeah.
Okay. Okay, so we're going to be talking a lot about two different types of lymphocytes, as you see here at the bottom of the slide, the so-called T and B cells, or T lymphocytes and B lymphocytes. So we can think of this adaptive specific defense as sort of a third line. You know, we talked about this metaphor of you being in a castle, if you will. And you have the moat out in front with the alligators. That's the first line of defense. You've got then that rock, 3-foot thick rock wall. If they get beyond the moat, the invaders have to get through the wall of the fort or the castle. Again, sort of a dumb analogy, but if you think of it in that way, this would be an additional line of defense. So they get through the moat, they get over the moat, they get through that first thick wall, and now you've got a second system in place to protect you. This is that third line of defense. And as I've been kind of reiterating a lot on the last few minutes, we're talking about protection against a specific pathogen. Again, it could be a bacterium, it could be a virus, it could be a protozoan, it could be any number of different items that we listed in that preceding slide about 5 minutes ago, right? Or it could be something that the pathogen secretes. When you guys take microbiology, some of you will in the nursing curriculum, we'll be talking about endotoxins and exotoxins that some bacteria produce. And it's the toxins that cause tissue damage to you and me. So our immune system may not necessarily target the cell that produces that toxin, but rather may target the toxin itself, because that's what's causing the problem. That's what's causing the tissue damage, the toxin. So if we can negate the toxin, the bacteria can't really hurt us, right? Because it's the toxin that it's using to cause the problem. And in other types of pathogens, it might be something that's part of the metabolic pathways that the cell produces along the way. If you remember way back, I don't know, what was it, the chapter 3 or 4, we talked about metabolism, and that was subdivided into catabolism and anabolism, if you remember. Catabolism was the breakdown of bigger molecules into smaller ones. Anabolism was the buildup of bigger molecules from smaller ones. So metabolism is a very broad term that describes all chemical reactions that are taking place within the cell. And sometimes it's those metabolic byproducts that trigger an immune response too. We generally will be kind of focusing on the pathogen itself, like the bacterial cell or the toxin that it produces, but it can vary. And what's really fundamental to this discussion we're going to have today is the notion that when we talk about B cells and T cells, and even this other cell you see listed here, macrophage, which is a monocyte-derived cell that's left the bloodstream and gone out into the tissues. That's what a macrophage is defined as. These important cells, B cells, T cells, macrophages, have the unique ability to distinguish between what we call self cells and non-self cells. So think of a self cell as being part of yourself, okay? Your bone cells, your muscle cells, your brain cells, your simple squamous of the endothelium of a blood vessel. You know, all of those cells that make up you are self cells. Non-self cells would be things like what?
Viruses and bacteria.
Well, viruses aren't technically cells, but they could be viewed as a non-self pathogen by the immune system. So you're absolutely right. I would include viruses in this group, but they're not technically cells. Okay, they're the acellular parasites. But bacteria is a good example of a non-self cell. A protozoan, a fungus that gets in, like a yeast infection, which is common in women, right? That would be viewed by the immune system, that yeast that causes the problem, as a non-self cell that needs to be removed. We would also include in this category of non-self cancer cells. Now, technically, where do cancer cells arise? Do you remember us talking about this back in chapter 4, I think it was?
It would be from a mutated self cell.
Exactly. Very, very good. It is a self cell that's gone haywire, right? Yeah. Uncontrolled cell division, all that good sort of stuff. So excellent. Our immune system, however, recognizes that cancer cell that has a very abnormal number of chromosomes and other cellular structures that would deem it non-self as something that needs to be neutralized and basically killed off, right? Okay. And the way that a B lymphocyte or a T lymphocyte would be able to distinguish, say, a cancer cell from another lymphocyte, let's say, is this unique array of protein receptors on the surface of the cell membrane. Now you got to go back and remember, we talked way, way, way back about cells and the plasma membrane. And if I asked you to describe to your mom what a plasma or cell membrane is made up of, what would you tell her in 2 sentences?
A plasma or cell membrane.
What is a cell membrane composed of? What are the parts to a cell membrane?
A phospholipid bilayer.
Yep, very good. That's a biggie. What else do you often find embedded in the bilayer?
Lipids and proteins, right?
You find lipids. Well, that's really what the phospholipid bilayer is made of, right? Phospholipid is a fat. So I don't know that I would list lipid because it's already there in the bilayer.
Okay.
What other big important organic compound? I think you mentioned it.
Proteins.
I'm sorry?
Proteins.
Proteins, right, and carbohydrates too. But proteins are a big component of that cell membrane. And I used the metaphor of an iceberg floating in an ocean of phospholipid. Do you remember me mentioning this? I did. So envision this ocean of phospholipid, because this is a fluid structure. This is not static. This is a liquid structure, this phospholipid bilayer. And then you've got this big iceberg protein bobbing around. Now, you know a lot about icebergs, right? Where's most of it? Potentially.
Underneath.
Exactly. You see just a small part potentially of the iceberg. So again, use your imagination. You might see a part of that protein on the surface, but a lot of it might be within the bilayer and extend down in toward the cytoplasm too. Okay, so it depends on the protein. Some are just on the surface. Some span the entire bilayer where there's a part on the surface and a part inside by the cytoplasm. And then there's even some where they're only on the inside part of the cell membrane. You can't see it from the outside. So there's all different kind of connections and permutations. But with regard to this arrow that I've got here on the screen, we're talking about surface protein receptors, okay, that are able to recognize chemically these foreign non-self cells or their secretions like toxins. And so we often use the term antigen to describe that portion or part of the pathogen that the protein receptor of the white cell being able to recognize that molecule, that antigen. And so we view these antigens as foreign molecules in our discussion here as being part of the bacterial cell or part of the virus or maybe something that the pathogen secretes, like a toxin. But to our T and B cells and macrophages, these are foreign antigens and they need to be neutralized. They need to be removed. And so from a chemical point of view, the antigen can take on a variety of different sort of signatures, if you will. A lot of times they're actual proteins that are part of the bacterial cell, maybe. But they can take on a lot of other potential molecular shapes, if you will, and compositions. Polysaccharides, of course, are sugars. Glycoproteins, glyco means sugar. So this is a partial sugar, partial protein molecule. Glycolipid means partial sugar, partial lipid, right? So they can take on a variety of different, chemical makeups, these antigens. Okay. Typically, they're fairly big in terms of their molecular size. And so they're fairly easy to recognize by our immune system, by our T cells and our B cells and so forth. Well, you remember from the blood chapter that when we talked about hematopoiesis, that this was the process by which the formed elements were made in the red bone marrow. Starting off at that hemocytoblast stem cell, right? And then it differentiated into either a myeloid or a lymphoid stem cell. Do you remember that flow chart? Yeah, go back and take a look at that. We're not going to review that here. I just want you to remember that ultimately that lymphoid stem cell is going to differentiate into lymphocytes. And of course, we're talking about the B and T cells here in this chapter. So these lymphocytes, which are about the same size as a red blood cell with a fairly large nucleus inside, they are produced in our red bone marrow, as are all of our blood cells, right? And the B cell, for the most part, matures in the red bone marrow, and then it will leave, as we can see here, and it will go out into various parts of the body, not the least of which is this important structure that we talked about, I think, recently, the lymph node. So what do we got coming in here?
Lymph?
Right, got that fluid coming in, the one side of the afferent components here. And then inside the lymph node, remember, we had those stationary B cells and T cells as well. And they're there to monitor the lymph for potential antigen, right? So these B cells are formed in the red bone marrow, and then they set up shop in different parts of the body, including the lymph nodes. But as it says here at the bottom too, you can find them in the spleen. Remember we said the spleen filters the blood, while the lymph nodes filter the lymph. The T cell also originates in the red bone marrow, just like the B cell does, but it generally travels to the thymus, which we talked about last week. Remember, this was located superior to the heart. This reached its maximum size when you were a kid, and it started to shrink as you got older. And it's here that the T cells mature, and that's where T comes from, thymus. Don't let that confuse you. They're really produced in their bone marrow, but they grow up and get their instruction booklet to become adult cells, okay, in the thymus. And then they leave the thymus, and they, like the B cell, travel throughout the body to set up shop, if you will, to monitor for antigen. And as we've said just a moment ago, some of those T cells will travel to the lymph node, others will travel to the spleen. I mean, they really travel all throughout the body. It's just not just those two structures, but all throughout the body, we find T and B cells. And they have those surface protein receptors, right, that can recognize antigen and do something about it. Note that the vast majority of lymphocytes here, upwards of 80%, are T lymphocytes, with the remaining 30 or so percent B cells. That's not to say that these are more important necessarily, but there is a slight skew, if you will, or a significant skew to more T cells than B cells on average in our bloodstream. They both have important roles to play, as we're going to talk about today. Okay, are there any questions so far? Okay. So we've said that these lymphocytes have these surface receptors that allow them to recognize the antigen, which is going to induce this immune response. But what's really amazing is when you look at how T and B cells are formed in the red bone marrow during hematopoiesis, that your bone marrow cranks out literally millions of varieties of both T and B cells. And what do I mean by millions of varieties? I mean that each of those T lymphocytes and B lymphocytes has a unique protein receptor. No two are alike. And if you're exposed, again, to a given antigen, virus, pollen, bacterium, you have somewhere in your body a particular B cell and T cell that has that specific receptor that will fit into that antigen. And we don't have time to really talk about how that occurs. It's quite a complicated process, but it involves genes being turned on and turned off. Gene regulation is really what it comes down to. And almost an infinite number of possible gene combinations that give rise to this enormous diversity of T and B cells. But just remember that you have somewhere in your bloodstream a particular T and B cell that has the proper shaped receptor that will recognize that antigen. Okay. So we're going to talk about T lymphocytes, and then we'll talk about B lymphocytes. We've said a moment ago that the T cells, like the B cells, form the red bone marrow, but that they get their instruction booklet, if you will, and grow up to be healthy T cells in the thymus, then they leave and go out into the body. Before a T cell can do anything in response to a foreign invader or one of its byproducts, like a toxin, it has to be activated, and that's the term they use, and therefore I put this word in red. It's a pretty important word. T cells must be activated before they can respond to that given antigen that they're able to recognize. And so the activation of T cells involves another type of cell that's going to present the antigen to that T cell. It can't do it by itself. It has to be presented the antigen. And as it mentions here, that antigen presenting cell, I'm going to show you a video on this in a moment, could be a close cousin, the B cell, or it could be a macrophage. Remember, this is defined as a monocyte that's left the bloodstream, undergone diapedesis, right, gone into the tissues, and there it becomes sort of morphed into this big, hungry cell. So macrophages can act as antigen-presenting cells. And once that happens, then the T cell is going to be able to do something in response to that antigen, but it can't unless or until it's been presented the antigen. I know it sounds kind of weird, but that's the process. So I'm going to go to this video and we'll come back to this slide. We might watch this actually a couple of times. It covers a lot of information, so don't let this overwhelm you, but just kind of sit back and see if you can glean a little bit of information about what an antigen presenting cell does and how a T cell works with that antigen presenting cell to recognize that antigen.
Th2 to stimulate. Proteins generally require the cooperation of
You can't see the video right now.
Th2
Okay.
to stimulate B cells into becoming antibody.
Okay, I always have a problem when I go to the video, so I need to stop, share, reshare. Okay, here we go. You guys see it now?
Yeah.
Okay, thank you.
Proteins generally require the cooperation of T helper cells, Th2, to stimulate B cells into becoming antibody-producing cells and memory cells. Such antigens are therefore said to be T cell dependent. The protein antigen is first ingested and then broken into peptide fragments by an antigen-presenting cell, such as a macrophage. The antigen is then moved to the surface of the macrophage on a class II MHC. The T cell receptor on the surface of a T helper cell recognizes the peptide antigen that is being presented on a class II MHC of the macrophage, and the T helper cell becomes activated. The activated Th2 cell is now capable of stimulating B cells.
Okay, I'm going to stop right here because I don't want to talk about B cells quite yet. But what I want to do, again, is just have you understand that here we've got an antigen presenting cell. That's this big guy in the middle. Let's pretend that this is a bacterium, okay? And this kind of spiky triangular shaped structure on the surface of the bacterial cell, let's pretend that is the antigen that makes that bacterial cell, again, a non-self cell, which this macrophage is going to identify as a non-self cell. And of course, what's it starting to do to that bacterium here?
Phagocytize it.
Exactly. It's phagocytizing it, isn't it? Yep. So it phagocytizes the bacterium. And then what it'll do is it'll break it down. It'll use its lysosomes, which remember are these other organelles containing powerful hydrolytic enzymes. And they'll dump those enzymes in there and it'll undergo what's called intracellular digestion. It'll just break that bacterium down. But what it then does is it takes the antigen, which was, remember, that little spiky triangular shape structure back on the surface of the cell here, right? It's going to take that antigen and put it on a particular type of receptor. It's called an MHC. That's what that blue pair structure is. It's a particular receptor, protein really. And on the tip of that MHC is the what? That's the antigen. Okay, that's the foreign antigen that was part of the bacterial cell, right? Everybody follow me? Okay. Here comes a type of T cell called a T helper cell. We'll be talking about a couple of different kinds of T cells, but that's what TH stands for, helper. It's going to assist or help in the immune response. And so it has to first be activated before it can help do anything. Well, how does it become activated? Well, it's got to interact with an antigen presenting cell. So if you look at the surface of this T helper cell, it has a special receptor that I mentioned a few moments ago that was able to recognize what?
The antigen.
A particular antigen, exactly, yeah. And in this case, this T helper cell is going to be able to recognize that particular antigen there in green. And how does it recognize it? Well, it actually comes into contact with the antigen presenting cell, and they can emerge right there. They're interacting with each other. The T helper cell then, once it's identified the antigen as presented by the macrophage here, then that T helper cell is said to be activated, and it can begin to stimulate B cells. And we'll talk about what B cell function is in just a few moments. But that's a really important function of T helper cells. They help the immune response. They must first be activated by an antigen presenting cell, and then they're going to release chemicals to basically cause this B cell to undergo cell division. And again, we're getting ahead of ourselves just a little bit, so I don't want to talk about that right now. But that B cell would not be able to respond in any way to the antigen unless the helper T cell assisted in that process. Questions on that? Clear as mud, huh?
The helper T cell, that's the only job that it has, right? Is to take the antigen, like read the antigen from the
Macrophage, yeah.
Yeah, from the macrophage, and then activate the B cells.
That's one important function that it has, yes. It releases chemicals called cytokines that do all sorts of different things, but one of the most important things that it does is it helps to activate and fire up the B cell.
Okay.
Yeah.
So it's almost basically a go-between.
Kind of, yeah, that's a good way to think of it. It is a go-between. But as you'll see in a few moments, it can do some other things as well besides just fire up the B cell. But it's absolutely in a go-between, yeah. it couldn't do this unless it was first presented the antigen by this antigen presenting cell. Yeah. Good questions. Anything else you want to ask about? Okay, I'm going to go back to the PowerPoint slide, I hope. Oh, let's see, where were we? I'm not sure if this was it. Yes. Okay. Upon recognition, T cells and some macrophages can synthesize and secrete cytokines, as I mentioned just a couple minutes ago, chemicals, special chemicals that can induce a whole bunch of different things, not the least of which is cause the B cells to get fired up. And we'll talk about some other cytokines here, and I think on the next slide. But the T cell would not be producing those cytokines unless it was first presented the antigen by that antigen presenting cell. And again, this MHC thing that they're talking about was nothing more than the protein that the antigen was part of, that was put on the surface of the antigen presenting cell. Okay, here are a list of some cytokines. These are chemicals produced by helper T cells upon activation by the antigen presenting cell. Colony stimulating factors causing the bone marrow to produce more lymphocytes. Why is that a good thing? Well, you're calling more troops to the battle, aren't you? Makes perfect sense. So more T cells, more B cells undergo hematopoiesis in the bone marrow. You're recruiting more soldiers and sailors to fight the war. Cause B cells to grow and mature. Well, we're going to talk about B cells in just a couple minutes, but without those cytokines produced by the activated T cell, they wouldn't be stimulated to grow and mature. So that's a critical role. Activates macrophages simply means get those big hungry phagocytic cells to get more active in the process. Here's interferons. We talked about it earlier in the class. Here they are coming in specific immune reactions to again prevent viral replication. Now remember, viruses must get into host cells where the host cell basically makes more viral particles, and then the virus is basically released by the host cell as it lyses. So this is targeting viral cells. This is targeting cancer cells. These interferons are extremely important chemicals that you're producing when you get sick or when you get cancer. Stimulates macrophages to engulf viruses. That's an important and good thing, right? We'll talk about B cells again in a couple minutes. One thing they do is produce things called antibodies. You've been hearing about that in the news, trying to take some of the antibodies that people have produced in response to being infected with COVID-19 and using those as a therapy for people who are sick, right? You've been watching the news, you've heard of this. Those are produced by B cells. Interleukins, another group of cytokines that assist in differentiation and growth of lymphocytes. This is that process that's taking place in the bone marrow. Again, calling more soldiers up, basically. Tumor necrosis factor, as the term kind of says, it's trying to kill tumor cells. So this tries targeting cancer. Also stimulates lymphocyte differentiation in the red bone marrow. Causes fever that accompanies bacterial infection. Yeah, did you know that fever has a definite role to play in fighting pathogen? Yeah, it does. When you get a fever, that means that your body is trying to up the temperature in your body, and that reduces the effectiveness of bacteria from reproducing, actually. It's real interesting. Okay, let's talk about a couple types of T cells. You've heard of these just a few moments ago, the helper T cells. As we said, become activated when the antigen presenting cell presents the antigen to the helper T cell. And if that helper T cell has the proper receptor that can fit into that antigen, then the T cell becomes activated. And as we said just a second ago, it can in turn produce a variety of different kinds of cytokines, all of which are calling more troops or activating their close cousin, the B cells. The whole role here, or the whole purpose here, is to fire up the system. We know antigen is there. What can we do to begin to address the presence of an antigen? Here's another type of T cell called cytotoxic T cells. These cells have a more direct role in killing antigenic, antigen presenting, or not antigen presenting cells. They kill cells that are pathogens like bacteria or virally infected cells or even cancer cells. So these guys are going to literally make contact with the cancer cell or the virally infected cell and kill them. So that's kind of what cytotoxic gets at. Let's watch a short video that talks about how cytotoxic T cells actually kill virally infected cells or cancer cells.
When a
Oh, wait a minute.
virus infects us,
You guys don't see this, do you?
No.
I'm going to reshare it, sorry. Let's try it again. We good?
Yep.
Okay.
when a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complexed with class 1 MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class 1 MHC. The cytotoxic T cell then releases cytotoxins, which induce apoptosis in the infected cell, and perforin, which causes perforations in the cell membrane. Self-proteins are also degraded, and the fragments presented on the surface via class 1 MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
Okay, let's watch that one more time. We'll turn off the narration here. Okay, so here, let me paint the picture here for you. This is, don't confuse this with an antigen presenting cell. This is not an antigen presenting cell in the sense of, you know, what we talked about a few minutes ago. This is a cell, one of your cells, one of your self cells that a virus is going to get into. Okay, as we said, viruses cannot complete their replication unless they get into a host cell, like this target cell. And then that target cell is going to end up making more virus. But in this particular case, when that virus sort of unwraps itself, and here we're looking at some viral nucleic acid, and we won't get into the details of this, but basically the viral DNA is initially transcribed into messenger RNA, and then it's translated into protein. We talked about that back in chapter 3. That process of how proteins are made, if you remember. So these yellow guys are viral proteins. This is made by the host cell, but it's a protein that's going to be used to help reconstruct eventually the virus. But what happens in this particular instance is the host cell takes a protein of its own. It's a class 1 MHC. Okay, now we talked about class 2 earlier. The antigen presenting cell used a slightly different protein receptor here. It looked blue and elongated, if you remember, but it's a different type of MHC. It's called this receptor. What's it got here? What is this? I know it says peptide fragment, but in our discussion, we would think of it as the what?
Antigen?
Right, the antigen. So here comes now that antigen that this cell is going to present on its surface. Okay. And guess who can recognize that antigen?
The helper T cell?
No. A cytotoxic T cell. Don't confuse them. Okay. This is a cytotoxic T cell. And the cytotoxic T cell has to have the proper receptor, as we've said before, to recognize the antigen, which it's going to do. There it goes, it recognizes it. Now, what is this cytotoxic T cell going to do in response to that recognition? It's going to kill the cell. it's going to release chemicals, one of which is called perforin, which perforates the cell membrane. So there goes perforin, and it cuts holes in the cell cytoplasm or cell membrane. Now this is one of your cells. This is not a bacterial cell. This is one of your cells that's been invaded by virus. And what eventually happens here is the cell dies. Well, you might say, well, gee, why do we want to kill that cell? Well, why do we want to kill that cell?
Is it so that the cell doesn't reproduce with the virus in it?
Right. We don't want more viruses to come out and then infect other cells. So if we have to kill this cell to prevent it from being a viral factory, then we need to do that.
So the cytotoxin T cell basically forces the cell to take one for the team.
Yes,
Okay.
that's exactly what it does. Yes, that's what it does. And it does so by releasing these powerful cytokines, not cytokines, but cytotoxins. Okay, they're chemicals that basically are toxic to the cell, kills the cell. Perforin pokes holes in the membrane and the cell cytoplasm leaks out. There it goes, the cell dies. So it takes one for the team, exactly right. Now what can this guy then do? Is he done? Well, he's done with the cell, but what can he do next? What can he do? He has to be able to kill other cells, but he can't kill other cells until what has to happen?
A virus gets into a cell again.
Yeah, exactly. And then he's got to go and recognize that same antigen, like he's doing here, right? And then he can release his chemicals and kill that cell. So he's going to be monitoring the area for more virally infected cells and kill them. Okay? So he's like special ops. Yeah. He's able to recognize that virally infected cell or that cancer cell and kill it. He doesn't want to kill other healthy cells that haven't been invaded by virus, does he? No. He's able to identify this non-self cell. Now again, it's non-self. Why?
They're mutated or has a pathogen.
Well, it's been invaded by virus, and that's what makes it, in the eyes of the cytotoxic T cell, a foreign cell that needs to be removed. Yeah.
So in diseases that have that like your body attacks itself, would that be something wrong with these cells?
Yes, exactly right. That's an autoimmune issue where the T cells here are attacking healthy cells. And that's a very involved, long discussion. But in essence, that's what's happening. It's something has gone haywire, whereby the immune cells are targeting healthy cells. Yeah.
Okay.
Yep. Okay, very good. Are we good with that?
Yeah.
Okay, you can go back and watch these videos, of course, as many times as you want. Oops, I didn't want that one, but we'll just go down here. Okay. And the last type of T cell that we want to talk about, in addition to helper T cells and cytotoxic T cells, are memory T cells. This unique ability, as we said earlier today, for our immune system to recognize that antigen should we be exposed to it later in life. If in 10 years you have that same bacterial infection or that same virus, we will have built up a memory, T cell memory, and those T cells will immediately differentiate into our friends, the cytotoxic T cells, that can begin to knock out those virally uninfected cells much, much faster because we know what to look for. We have those special receptors on our surface that cytotoxic T cells do, or will have, to recognize that antigen much, much quicker. These memory T cells divide and differentiate into cytotoxic T cells. The memory T cells themselves, they can't attack cells. They can't do anything. They can differentiate into cells that do that.
So Are the memory T cells like essentially an antibody?
No,
Is that what they're kind of looking for?
no, nope, they have nothing to do with the
No.
antibody. Nothing to do with the antibody. They are simply there circulating in your body such that if you are exposed to that same antigen down the road, those memory T cells will recognize that antigen and then form into cytotoxic T cells, which will kill the virally infected cell or whatever it might be.
Okay, so like with COVID-19 then, they're saying that there's a chance that you could be reinfected with it. So how does that differ from any other virus?
I cannot speak to that, Sarah. I do not know enough, nor have I really researched it. But I understand your question, and it's a very good question. I don't know.
Okay.
I'm not a virologist. And yeah, I mean, there are answers to some of those questions, but I'm not even sure that we know today why some people might have, lifelong protection while others may not. Generally, there's protection or some degree of protection, but now they're saying there's no guarantee if you've been subjected to COVID-19 that you couldn't become reinfected.
Mm-hmm.
And therefore, if you're becoming reinfected, you could become or continue to be a carrier, right? That's the real concern too. Okay, so when we talk about memory cells and cytotoxic cells and helper T cells, we're talking about something called cell-mediated immunity, or CMI. So associate cell-mediated immunity with T cells. And the B cells, which we're going to talk about next, are involved with antibody-mediated immunity, or AMI, antibody-mediated immunity. So the antibodies that you're hearing about in the news today about people having the COVID-19 antibodies, those antibodies were produced by B cells. So like the T cells, we have B cells circulating in our body that have special receptors on their surface that are able to identify specific antigen. And here we have two different kinds of B cells here in terms of their protein receptors, right? If you take a look, you see these guys have the green colored tips and these guys have the orange colored tips. They don't really have green and orange tips, but we're going to use that as a diagram. Could this cell here recognize those antigens on the surface of this bacterial cell? Could these fit into there?
No.
No. So this B cell is oblivious to the presence of this bacterium. It can't recognize it. It can't do anything about it. But what about this cell? It, of course, has the receptor, right, that can fit into the antigen as it's done here. Okay? Now, that alone doesn't activate the B cell. It's an important process that has to happen, okay? But look what we have going on here. We have a T helper cell that just recognized the antigen presented to it by the antigen presenting cell. And as we said, in response to that, one thing that helper T cells do is release cytokines, different kinds of cytokines, right? Like that we talked about in that table. Well, these cytokines here are going to take that B cell that has recognized the antigen and fire it up and activate it. And so here is the activated B cell, which would not have become activated in the absence of the cytokines produced by the helper T cells. So what I want you to start to begin to see here is that the B cells can't do it alone. And as you're going to see, the T cells can't do it alone. They have to work together. What does the activated B cell do? Well, it does a bunch of different neat things. One thing that it does is it undergoes cell division. So to review, here's our activated B cell. It's connected to and identified the virus or the antigen on the surface of the bacterial cell, whatever it might be. And here are the cytokines that were secreted by the activated helper T cell. Gets this cell really fired up. It undergoes cell division. It undergoes what's called clonal expansion. Basically, you make a whole bunch of copies of that cell. How do I know these are copies? They all have what?
The same receptors.
Right, they all have the same receptor. They are all now able to recognize that antigen. Not only that, but look what happens to these cloned B cells. They undergo further division and differentiation, and some of those will form into what are called plasma cells. Now, those are these two guys. Plasma cells actually secrete antibodies against the antigen. So these are like little missiles that get sent out by the plasma cells. And then the other type of cell that's produced here, of course, you see in yellow, memory B cells, not to be confused with memory T cells. The memory B cells are there for what reason?
To hold on to fight later on.
Right. If you're subjected to that same antigen 10 years from now, these memory B cells will differentiate into plasma cells that will release antibodies. Okay, so they're just there to remember the antigen. And if they see it again, they will differentiate into a cell that can do something about it. In this case, the plasma cells are secreting these things called antibodies. And they produce lots and lots of antibodies. This interesting statistic says that at the peak of infection, each one of those single plasma cells can crank out 2,000 antibody molecules per second. So you're flooding your lymphatic system and your blood stream and everywhere you've got an infection with tons and tons of antibodies specific to that antigen. This simply reviews sort of what we just spent the last 15 minutes talking about. So I'll just let you read that over. What is an antibody or immunoglobulin? Sometimes that's the word that's used in some textbooks. Immunoglobulin is kind of a mouthful, IG for short. It's another name for antibody. So this is a protein produced by those plasma cells. And it's here on the sort of the tips of this molecule. They don't really look like Y-shaped structures, but we're using a model sort of to depict a protein. This is where the antibody binds with the antigen. There's a physical connection that occurs between the antigen and the antibody. And there's what they call here variable and constant regions. I'm not going to get into all that now, but there's a wide diversity of different kinds of antigen binding sites that can be produced by our plasma cells, because there are a wide variety of antigens that need to be identified and connected to the antibody. So we've got 5 major classes of antibodies. They're listed here. IG, remember, stands for immunoglobulin. So there's IgG, IgA, M, D, and E. Those are the, I'm not sure where the letters come from, but that's how we designate these from one another. And your immune system will produce these different antibodies in response to different types of pathogens you might be exposed to. As you see here, IgG is produced when you have a bacterial infection or a viral infection or exotoxins that are being released by certain bacteria. Notice it says here too, IgG also activates complement. We talked about complement back earlier in the chapter as part of nonspecific innate defense. The vast majority of antibodies produced by our plasma cells are of this category or class IgG. And then there's, again, 4 other types too that are produced in response to other things. Most all of these antibodies have sort of a Y shape to them. When you look at sort of their modeling, we won't really get into the difference. You don't have to differentiate these, so don't worry about, you know, this table. But there are the 5 different classes, and some of these are monomeric. There's 2 antigen binding sites, like you see here, and here's some that have like 5 different monomers, or a pentamer is the term we use. 5 of these little Y-shaped arms. But it's here that the antibody will connect to the antigen at the tips of these little Y-shaped things. Well, what happens when an antibody comes into contact with an antigen? And that's the next thing we want to talk about. Because remember, what's the goal here? The goal is to rid the body of what?
Antigen.
The antigen, right? Which could be part of the bacterial cell. It could be a toxin produced by the bacterial cell. It could be a surface marker of a virally infected cell. It could be a surface marker of a cancer cell. We got to be able to identify the antigen or the antigen-bearing cell and kill it. How does that happen? Well, one way that that happens is via this direct attack process. When the antigen and the antibodies actually physically combine together, it can lead to easier identification of that antigen by white cells. Phagocytic neutrophils and macrophages can gobble those marked cells. Okay, so envision a bacterial cell now with all these antigens sticking out, and now you got antibodies connected to it. Well, it's going to mark that cell for easier identification. Yeah, that's what's going on here. So these are effects of that direct interaction between antigens and antibodies. The complement system we talked about again last week. This was a bunch of proteins that had a variety of roles to play. Again, these are reviewed here. All of these can be set into motion when there's antibody-antigen interaction. I used the silly metaphor last week about taking an M&M and coating it with chocolate. Remember my peanut M&M analogy? Mm-hmm. Yeah, so if we can coat the pathogen with complement proteins in the same way that we can coat the pathogen with antibodies, it makes it easier for a phagocytic cell to identify that cell as foreign and one that needs to be neutralized. So complement can coat cells. Complement can act as a chemotactic mechanism, like dumping the blood off the back of the boat if you want to catch sharks. Remember that metaphor? So this is attracting basically phagocytic cells to that injury site, to that infection site, gobble up the bacterial cells, for example. So there's just a whole host of important effects that complement can have. But again, this is set into motion when antibodies combine with the antigens. And inflammation was another thing that we reviewed back in that table of innate defense. This is the redness, the soreness, the pain, the swelling. We've all suffered inflammation at some point in our lives. Well, sometimes inflammation can result from antibody-antigen interaction as well. I'm going to let you watch this video. It's a great summary of what we've just spent the last hour or so talking about. I want to talk, as we wind down the chapter now, about something called a primary and secondary immune response. When you are exposed to, let's say, a given antigen, let's say somebody, sneezes on you and you inhale the virus and you get the flu. Or you get a sliver and introduced into that sliver were some bacteria that cause an infection in your finger, something like that. If you could monitor the antibody levels in your bloodstream, okay, so here's when you got the infection right there, day 0. You'd find if you could monitor the blood antibody titer, it's called, or antibody concentration, you'd find that the antibody levels take a while before they get high. Now they're using the example here of a better part of almost 2 weeks before you reach peak antibody levels. And then generally the amount of antibody begins to tail off and eventually will go down to 0 as you get over that infection. Okay. But what happens if you are subjected to that same antigen in, say, 3 years? What do you think you're going to see here in terms of an antibody reaction or response?
It should peak again because
well, it's going to peak again, and look what happens. It's not only peaking, but look at the timetable. I'll flip back and forth here. It took a while to get maximum high antibody levels, right? But a second time around, wow, boom, within less than a week, you are extremely high, higher than you were here in terms of concentration, but the point is much faster and lasting longer. And that's due to the presence of those memory B cells that differentiated into plasma cells, right, that produce the antibodies. So you already built up a memory to that, and you reacted to that much, much quicker. Not only that, but remember we have memory T cells that differentiate into cytotoxic T cells that can knock out cells as well. Now, this isn't monitoring that process. It's looking at strictly antibodies, but those memory cells are extremely important, aren't they? This is reiterating the importance of memory B cells in this secondary response, which is much, much faster. That's why you're not going to get sick here. You're going to nip that in the bud before you even get an infection, probably. You might not even know you were exposed to that antigen. So let's talk about some classifications of immunity. Naturally acquired active immunity. This is what happens when you get an infection from, again, inhaling virus that somebody sneezed into the room air that you walked into. This is the cut you got that introduced the virus or the bacterium into your bloodstream, and you got an infection as a result of that. You got it naturally, and you built up active memory to that. In other words, B cells, memory B cells, memory T cells are going to be made. You're going to fight the battle. You're going to have to target that particular antigen and get rid of it the first time around. But you've got built-in protection, so if you get it later, you can knock it out quicker. How about naturally acquired passive immunity? Any idea what that might be? Naturally acquired passive immunity.
Wouldn't that be like the antibodies that you get from like mom's milk and stuff?
Right, exactly. If mom breastfeeds the baby, she is providing immunoglobulins, antibodies, to the baby via the colostrum, via the breast milk. Yeah, it's natural, right? Breastfeeding is a natural process. And the baby is getting the antibodies, which provides more short-term protection, but nonetheless protection to a given antigen. Now, will the baby build up any sort of memory?
No.
No, no, no memory. But the baby's provided protection. And that's why it's so unique to me anyway. It's amazing how long babies stay healthy. You know, for the first 6 to 8, 10, 12 months, you don't see babies generally getting sick. And that's because not only with the baby potentially being breastfed help in that process, but during the pregnancy, there are immunoglobulins, antibodies that cross the placenta. And so mom gives it to the baby just in utero as the baby's developing as a fetus. Yeah. Artificially acquired active. Well, what does artificially acquired kind of get at?
Like a vaccine or something?
Right, you're getting a shot. You're getting some sort of fluid in you via, you know, a needle. What does active immunity kind of mean?
Live.
I'm sorry?
Does it mean like it's live?
Well, it could be live, yes, but that doesn't mean active immunity.
That you're acquiring memory?
Say that one more time.
You're acquiring the memory cells through it?
Exactly. The end result will be the formation of memory T and B cells. Do you have to fire up the helper T cells? Do you have to have helper T cells secreting cytokines? Do you have to have all that activity taking place? The answer is yes. You have to go through that to build up memory. You have to sort of fight the war. However, do you want the patient to get sick with fever and nausea and vomiting and diarrhea?
No.
No, of course not, no. But you want to trick the immune system into thinking you got the antigen. And you did via the vaccination. So when you get a shot, for example, a vaccine, it could have in it what's called an attenuated virus, a weakened form of the virus. Weakened in the sense of not making you sick, but tricking the immune system into thinking you got the real McCoy, and therefore taking steps to build up immunity to that. So you're tricking the immune system, really. Why would you want to do that?
So you don't actually get like full-blown sick from it?
Okay, you must assume you're getting that vaccination because you run the risk down the road of naturally acquiring that antigen.
Yeah.
Right. And if you do, what will you have at your disposal to fight it off?
Or antibodies?
Well,
Memory.
memory, that's the key. You have memory, which could result in antibodies being made. You're right, Sarah. But the key is you have built up memory to that antigen.
Okay.
That's the key. And our last one, artificially acquired passive immunity. What do you think this is? Whenever you see artificially acquired, think what?
Shot.
Right, shot. What am I going to shoot you with? The attenuated, weakened form of the virus? No, that's number 3.
A dead form of the virus?
No, good guess.
Well,
We just talked about this possibility not 10 minutes ago.
would this be like what they're doing with like the COVID-19, taking the antibodies?
Yes.
Okay.
Yes, this is giving the antibodies directly.
So people who would be receiving the antibodies, it can help them fight it now, but it won't help them fight it down the road.
Exactly.
Okay.
The idea is give them immediate protection, short term, but immediate, because there's a chance that if we don't give them the antibodies right away, that it might be too late to let them go through the artificially acquired active, because there's a time frame here you got to be aware of. In order to build up memory, it doesn't happen overnight. It takes weeks and weeks to do that. And you might not have weeks and weeks to, at your disposal. You might need to give immediate protection to those senior citizens in the nursing home, right? So if you can give them the antibody today, they're protected today. That's the upshot. The downside is their immune system is not going to recognize that antigen if you're exposed to it, later in life. But that may not be an issue because, grandma's 92. Let's not worry about down the road. Let's get her through the next week. You know what I'm saying? So there's a table here that I thought just did a very nice job, reviewing those 4 different types or categories, and so you can just kind of check that out too. It's what we just talked about. So in review, as we said earlier, we've got the test coming up on Thursday, 100 points over chapter 15 and 16. I will be getting the grades in on Friday. So it's going to take me, a day to correct the exams and crunch the numbers and do all that. So my plan is to have those grades submitted by the end of the day Friday. And then I was talking to the registrar the other day, and she was saying that once she gets all the grades in, she then will free those up in Banner, so you can go in and check all of your course grades. I think like probably later in the day Friday, or certainly by the end of the weekend, all of the grades should be in. I'm going to get mine in by the end of the day Friday. I will also post in Blackboard an Excel spreadsheet with all of your J numbers, the last 4 digits. I'll give you the exam 4 score. If you did an extra credit paper, I'll list that, and I'll give you a total accumulated total points for the course, all in that Excel spreadsheet, so you can take a look at that too. Okay. Any
So what's the
questions?
potential total of the whole?
Say that one more time.
What's like the potential total? Like if say you got 100 on everything.
I have here 6. 690. I had sent out earlier in our remote learning a revised, upgraded, updated summary because we had eliminated the lecture quizzes, but we kept the lab quizzes. So I'm coming up with 690 possible total points, excluding the extra credit paper. And by the way, I think this is also posted in Blackboard. Okay, so you can go to that anytime you want. All of the points by lab and lecture are listed, as are the percentages and total points by grade. Okay, that's all down in there. If you have questions, just let me know. All right. Kind of went over a little bit. Any questions on stuff we talked about today?
I think so.
I'm good.
Okay, well, I hope you guys all have a really great summer. I know it's been a very strange, you know, what, 8 weeks almost, I think it's been. In some ways, it seems like it's been a long time, and in other ways, it seems like it hasn't been. Maybe are we getting used to this a little bit, maybe? I don't know, a little bit. I don't prefer it over face-to-face. I miss seeing you guys, but I wish you the best of luck. I'll probably see you around, hopefully in the fall, because a lot of you guys are taking A&P 2, I assume. I don't know if we'll be back face-to-face, you know, that's all up in the air still. I hope so, but I just don't know. We seem to be phasing in this stuff in New York State a little bit slower. I'm not saying that's a bad thing, but I think we're in like tier 4 of the 4 tiers in terms of, you know, phasing back into normality. So education is like the last group, unfortunately, which I understand, but. So anyway, I hope you have a good summer. Thanks for hanging in there, and I will see you next semester, I guess.
Okay. Thank you.
You bet.
Thank you.
Thank you.
You bet, guys. See you later.
Bye.
Bye.